Production of cabbage, Brassica Oleracea L. as affected by humic acid and complex fertilizer NPK. Abstract. This study was conducted at the Agricultural Research Farm in Hada, Al-Sham of King Abdulaziz University in Jeddah in 2018/2019 and 2019/2020 to evaluate the effect of humic acid fertilizer, 0, 50, 100 liters/hectare, and NPK ferlitzer, 0, 600, 900 kg/ha, and their interaction on the growth and yield components of the cabbage plant, Brassica Oleracea L. All the different treatments witnessed high significant increases, p equals 0.01, in the morphological characteristics, stem length, stem diameter, head length, head diameter, head roundness coefficient, and in the properties of leaf area index and in total yield. The percentages increased with increasing the rate of humic acid from 0 to 100 liters/hectare, and with increase of NPK from 0 to 900 kg/ha. The results of this study recommend fertilizing cabbage plants with HA at a rate of up to 100 liters/hectare, and with NPK fertilizer at rate up to 900 kg/ha, and also recommend application of mixture of HA and MPK fertilizers at rate of 50 L/ha with 900 kg/ha for optimum cabbage yield, and 100 liters/ha with 900 kg/ha NPK for other cabbage growth parameters. Keywords: humic acid, Ha, cabbage, Brassica Oleracea L, NPK fertilizer. Introduction. The cabbage plant, Brassica Oleracea var. Capitata L, which belongs to the Crusader family, Brassicaceae, is one of the families that have high economic importance, and is of high importance for the Saudi consumer, and it is one of the imported vegetables, and its cultivation is badly needed in the kingdom to improve its production conditions. Agriculture in the Kingdom of Saudi Arabia has become increasingly important, especially with the current limited water supply and the increasing global demand for food. Agriculture in the kingdom mainly requires looking for factors that enable plants to obtain water and nutrients, hence the importance of using fertilizers and mineral and organic compounds to enhance growth and productivity. The use and application of compounds or biostimulants that promote growth are a promising tool for increasing the growth and productivity of plants in general, including vegetable crops, especially in light of harmful environmental and climatic conditions. Humic acid increases the growth and productivity of various crops, including vegetables, due to their effect on soil properties, soil water availability to plants, as well as nutrients. Atia et al. 2002, Zandonati, et al. 2007. One of the many properties of humic acid is its positive effect on nutrient uptake by vegetable crops. Samir and Yilmaz, 2005, Zandonati et al. 2007. Humic acid is a promising natural resource to be used as a fertilizer alternative to increase crop yield. Nickbacked et al. 2008. Humic acid contains many elements that improve soil fertility, increase the abundance of nutrients, and thus increase plant growth and production through stimulation of cellular respiration, photosynthesis, water and nutrient uptake, and enzymatic activities. Piccolo, 1996, Chen et al. 2004. It also improves soil physical properties, controlling hormone levels, thus promoting plant growth. Ciernella et al. 2002. And many researchers have proven the positive effects of humus, HA, on the growth and productivity of many plants. Atia et al. 2002, Dursun et al. 2002, Turkmen et al. 2004. Plant growth, development, and yield increase are directly affected by humus acid. It improves plant absorption of nutrients, leads to availability of minerals to the plant, improves plant growth and controls soil-borne diseases. Moro Mikhail et al. 2011. Fertilizing with chemical, NPK, or organic fertilizers increased the available total phosphorus content, making it more available for plants. Huang et al. 2016, Shi et al. 2014. Application of the chemical fertilization, NPK, enabled growing rice twice in the same land during 1 year. Yat, et al. 2015. Addition of the chemical fertilizer NPK to soil growing 2 species of bell flower resulted in increase of plant length, leaf area, number of nodes and number of branches. Quan, et al. 2019. The aim of this research is to study the effect of humic acid as an organic fertilizer and NPK as chemical fertilizer on stimulation of growth and yield components of cabbage, Brassica Oleracea L. Materials and methods. The research was conducted at the Agricultural Research Station, King Abdulaziz University, Hada Al-Sham during 2 agricultural seasons, 2019/2020, and 2020/2021. Randomly split plot design with 3 replicates was used. The main plot treatments were 3 concentrations of humic acid, 0, 50, and 100 L/Ha, and the subplots were 3 concentrations of NPK, 0, 600, 900 kg/Ha. Cabbage was grown in rows in plots of 3x3 square meters. The distance between the rows is 50 centimeters and the distance between the plants is 50 centimeters. Agricultural operations. Land preparation for planting. The soil was plowed with a plow to a depth of 25 to 30 centimeters and then leveled. The area was divided into plots of 3x3 meters distributed according to the design of the experiment. Planting methods. During the first season, seeds were sown in 26-11-2019, and seedlings were planted in the field in 25-12-2019, and harvest was conducted in 17-2-2020. During the second season, seeds were sown in February 11, 2020, and seedlings were planted in the field in April 12, 2020, and harvest was conducted in March 3, 2021. Irrigation system and networks. A drip irrigation system was used, containing a 6000L storage tank, disc filter, pump, control and solenoid valve was used to control the flow time. Irrigation was carried out daily based on the full water requirements of the cabbage crop. The site was plowed and leveled, and the drip lines were placed at a distance of 50 centimeters between the lines and the distance between the points was 50 centimeters. A 125 m disc filter system was used. The required irrigation water was calculated based on the requirements for crop need, evapotranspiration, and the total available water in the soil. Experimental data. 5 plants were randomly selected at harvest for each treatment unit and morphological characteristics were measured including stem length, stem diameter, head length, head diameter, coefficient of roundness, leaf area index, as well as the total yield weight of the crop in ton/HA. Statistical analysis. Analysis of variance for complete block design was used. Significant differences between means of the different treatments were statistical determined using the LSD test at p0.05 according to El-Nakhlai, 2010, with the use of the SAS program for statistical analysis SAS, 200. 2 results and discussion. The data will be presented by analyzing the contrast between the different treatments of humic acid and different treatments of NPK and discussing the averages of the different morphological characteristics and the components of growth and yield of the cabbage plant, Brassica Oleracea var. Capitata L, resulting from these treatments. The data will be presented by analyzing the contrast between the different treatments of humic acid and different treatments of NPK and discussing the averages of the different morphological characteristics and the components of growth and yield of the cabbage plant, Brassica Oleracea var. Capitata L, resulting from these treatments. Morphological traits. Analysis of variance. There are high significant differences, p = 0.01, between the different treatments of humic acid and NPK on stem length, stem weight, and head diameter of cabbage plants during the first and second agricultural seasons, Table 1. Asterisk indicates significant difference at p = 0.01, level, significant difference at p = 0.05, level, NS indicates no significant difference. Averages. Length and diameter of stem and head roundness. Table 2. Indicates significant average increases in stem length and diameter, head length and diameter, and head roundness of cabbage plant under HA application by 23.85, 13.90, 29.12, 26.42%, respectively under 50 L/Ha, and by 48.63, 14.15, 58.89, 53.40%, respectively at 100 L/Ha fertilizer during the first season with no significant difference in head roundness. Under NPK fertilizer the parameters increased by 13.85, 14.12, 1.49, 52.39, 2.16%, respectively at 600 kg/Ha, and at percentages of 25.43, 29.17, 39.36, 71.85, 2.77%, respectively at 900 kg/Ha during the first season. Also the averages of the studied parameters were significantly different under both HA and NPK fertilizer rates during the second season, Table 2. Table 2. averages of the morphological characteristics of the cabbage plant parts, stem length, stem diameter, head diameter, and roundness coefficient under the influence of different rates of humic acid and NPK for the 2 seasons 2019/2020 and 2020/2021. Second season. First season. The averages followed by the same letters did not differ significantly from each other according to the LSD test at 0.05 significance level. The effect of interaction between humic acid and NPK fertilizer rates on the morphological characteristics of the cabbage plant. The results of the averages, Table 3, indicate significant effects of the interaction of HA and NPK at all their different rates resulted in differences in the averages of stem length, stem diameter, head diameter, and rotation coefficient. The combination 100L/HA with 900 kg/HA NPK resulted in the maximum percentage increases in stem and head characters. Stem attained the highest percentage increase 27.27% in length, 38.30% in diameter, and head attained 48.16% in length, and 71.45% in diameter. While the combination 50L/HA with 900 kg/HA NPK gave the highest percentage increase in head roundness, 52.77%. Each type of fertilizer has its own effect on crop growth and crop production due to the different nature and characteristics of these fertilizers in releasing inorganic and organic nutrients. The humic acid and NPK fertilizers led to significant increases in the morphological characteristics of the cabbage plant, length and diameter of the stem, the length and diameter of the head, and head roundness coefficient. These results correspond to many of the results reached by a number of researchers. Baldotto et al., 2009, and Chen et al., 2004, obtained significant increases in plant growth and the growth of their roots and stems with the addition of humic acid fertilizer. Soar, et al., 2018, also found significant increases in the height and diameter of the head of the cabbage plant when fertilizing the plants with humic acid fertilizer. Al-Dabak, et al., 2020, found a significant increase in the length of the stem of the grapevine when fertilizing with humic acid fertilizer. Also, Sani, et al. 2015, found a significant increase in the height of canola plants when fertilized with humic acid fertilizer. Fan, et al. 2014, found a significant increase in stem diameter and head diameter of chrysanthemums when fertilized with humic acid fertilizer. Hope, et al. 2016, found a significant increase in head circumference and diameter of grapevine after fertilization with NPK fertilizer. Loxano and Shugino, 2019, obtained significant increases in plant height, stem diameter, and root width of grapevine when fertilized with NPK fertilizer. Fan, et al. 2014, found a significant increase in stem diameter and head diameter of chrysanthemums when fertilized with humic acid fertilizer, as well as with NPK fertilizer. Patel, 2010, fertilized the cabbage plant with NPK fertilizer with an average 75-100-150, and obtained a significant increase in plant height, head length, and head diameter. Moy and Yezu, 2015, added NPK 15-15-15 fertilizer at a rate of 300 kg/HA to the grapevine plant and found a significant increase in plant height, stem diameter, number of leaves, head length, and head diameter. When HA and NPK fertilizers were combined, this interaction led to significant increases in cabbage plant characteristics compared to their individual effects. These positive effects of the combined interaction of humus fertilizer and the chemical NPK on cabbage morphological characteristics are consistent with many previous studies. Verma, et al., 2014, found a significant increase in the height of the grapevine when fertilized with a mixture of humic acid and NPK. When Islam, et al. 2017, fertilized the grapevine with organic fertilizer mixed with chemical fertilizer, they obtained a significant increase in plant length, head length, and stem length. Analysis of variance. There are high significant differences, p = 0.01, between the different treatments of humic acid, and different treatments of NPK on leaf area index, and yield, ton/hectare, during the first and second agricultural seasons. Table 4 indicates significant difference at p = 0.01 level, asterisk indicates significant difference at p = 0.05 level, ns indicates no significant. Averages. Averages, ton/ha, leaf area index and cabbage yield leaf area. The means of leaf area, leaf area index and cabbage yield were significantly increased by the addition of humic acid and NPK fertilizers, Table 5. The increases reached 106.66, 76.00, 79.18%, under 50 L/HA fertilizer, and by 198.33, 150.00, 163.02%, at a rate of 100 liters/hectare respectively during the first season. Also the means of leaf area index and cabbage yield were significantly increased by the addition of the chemical fertilizer NPK, up to 46.91, 45.34, 65.47%, respectively under addition of 600 kg/ha, and by 100.00, 98.30, 156.5%, respectively under application of 900 kg/ha during the first season. These studied parameters were also significantly increased with application of both HA and NPK fertilizers. Leaf area index. The averages followed by the same letters do not differ significantly from each other according to the LSD test at 0.05 significance level. Interaction effects of HA and NPK rates on leaf area index and yield. The results of the interaction of HA and NPK, Table 6, showed increases in averages of leaf area index and cabbage yield with significant differences in these parameters. The optimum combination that gave the highest percentage increases in leaf area and leaf area index was combination 100 L/HA with 900 kg/ha NPK resulted in the maximum percentage increases in leaf area, 105.78%, during the first year season. While the combination 50 L/HA with 900 kg/ha NPK was the optimum combination for cabbage total yield giving the highest percentage increase, 195.00%, during the first season. It is evident from these results that the joint interaction between the different rates of humus acid, 50 and 100 liters/hectare, and the rates of NPK fertilizer 600 and 900 kg/ha, led to significant increases in leaf area index and yield of cabbage plant, and the percentages increased with the increase in the rates of HA and NPK. The results of this study demonstrated the significant effect of humic acid and NPK fertilizers on the characteristics of cabbage leaf area index and yield. The application of these fertilizers led to significant increases in averages of these characteristics during the two planting seasons compared to control. These fertilizers' effects on cabbage characteristics are consistent with many previous studies. Irrigation et al., 2013, found a significant increase in leaf area and biomass of maize leaves when adding humic acid. Abu al-Hassan and Al-Sham, 2015, found a significant increase in the components of red grape production when fertilized with humic acid and NPK. Ahmed, et al., 2013, fertilized Gladiolus grandiflorus with humic acid and obtained increases in foliage growth per plant, leaf area, stem diameter, and length. Bedani et al., 2016, added 3 rates of humic acid fertilizer and found a significant increase in the production and quality of onions with the increase in the acid rate. Lee, et al., 2019, found that the use of humic acid over 3 years improved the yield and quality of the peanut crop. The increase in crop productivity due to the addition of humic acid was observed in many crops, including tomatoes and potatoes, Grady and Tina, 1999, onions, Eric et al., 2000, and maize, Irrigation et al., 2013. Soar, et al., 2018, found a significant increase in cabbage production and yield quality when fertilized with humic acid fertilizer. Also, Al-Dabak, et al. 2020, found a significant increase in cabbage yield after fertilizing with humic acid fertilizer. Al-Ubaidi, et al., 2019, treated red cabbage plants by humic acid fertilizer at a rate of 0.5.10 ml/liter and obtained significant increase in the growth and yield components compared to the control treatment. Chen, et al., 2017, obtained significant increase, 29.56%, in the yield of sweet potato under fertilization with HA plus N. Patel, 2010, fertilized Takrambe with NPK fertilizer, 75-100-150, and found a significant increase in the production of plant heads. In a study carried out by Miaj et al., 2011, the fruit and yield of Solanum melangina reached twice the weight when fertilized with NPK fertilizer, and NPK fertilizer at the rate of 200 kg/ha improved pod production, weight of 100 SEDS, pod weight and final grain yield per hectare in cow P, Kareem, ETAL, 2013, and there was a significant increase in wheat production, Jing, ETAL 2015. Duran and Banyoko, 2003, found significant increase in onion production with NPK fertilizer. Moy and Yezu, 2015, added NPK fertilizer, 15-15-15, at a rate of 300 kg/HA to the grapevine and found a significant increase in leaf area and production components. Humic acid provides nutrients to plants because it controls the exchange of carbon and oxygen between the soil and the atmosphere, which stimulates and increases plant growth. Baransikova and Makovnikova, 2003, Calvo ETAL, 2014. Turkmen, ETAL, 2004, assumed that humic acid influences plant yield through its effect on respiration and photosynthesis. Moro Mikhail ETAL, 2011, says that humic acid is an organic substance whose role lies in resisting diseases in the soil, improving soil properties and absorbing nutrients, and thus significantly affects plant growth. It was shown by Mart, 2007, Samir ETAL, 2005, that humic acid improves soil properties, increases enzyme activity, and increases crop productivity. Also, many studies have proven that adding humic acid to the soil helps to improve the absorption of elements by plants. Makowiak ETAL, 2001, Sharif ETAL, 2004. Lee, ETAL, 2019, found that adding humic acid to the soil over a period of 3 years improved the physico-chemical properties, enzymatic activities, and microbial diversity of the soil, which helped alleviate the obstacles to continuous peanut cultivation. Humic acid acts as a pH regulator, Campitelli ETAL, 2008. It also has the benefits of holding ions and works like chelating materials, Pinheiro ETAL, 2007, Manzoor ETAL, 2014. NPK enhances plant growth because it is quickly absorbed by plant roots, Xiao Yuan Yan, 2010. Stefano ETAL, 2004, reported that chemical fertilizers have a strong effect on crop growth, development, and yield. Also, Fashina ETAL, 2002, indicated that providing adequate nutrients from chemical fertilizer leads to improved cell activities, reproduction, and growth. To conclusion. This study was conducted to evaluate the effect of humic acid and NPK fertilizer on the growth and yield components of cabbage, Brassica Oleracea L, in 2018/2019 and 2019/2020. All the different treatments witnessed high significant increases, p equals 0.01, in the morphological characteristics, stem length, stem diameter, head length, head diameter, head roundness coefficient, and in the characteristics of leaf area index and in yield production. The percentages of the increases increased with increasing the humic acid rate from 0 to 100 liters/hectare. An increase in NPK from 0 to 900 kg/ha, and also in the interaction treatments of HA with NPK. The results of this study recommend adding humic acid to the soil on which cabbage plants are grown at a rate of up to 100 liters/hectare, and applying NPK fertilizer at rate up to 900 kg/ha because these amounts lead to significant increases in cabbage plant growth components and significant increases in cabbage plant yield production components. It also recommends application of mixture of HA and MPK fertilizers at rate of 50 L/ha with 900 kg/ha for optimum cabbage yield, and 100 liters/ha with 900 kg/ha NPK for other cabbage growth parameters.